What if I told you that changing just one functional group or one ring in your molecule made the difference between a failed campaign and a billion-dollar blockbuster drug? Today, I'm revealing the chemistry trick that big pharma and agrochemical R&D departments use every day. And if you stick around and reach out, I'll share with you some Python code to do it yourself. Hey folks, welcome back. I'm Dr. Avery. Today we're going to be talking about bioisostere replacements. A bioisostere replacement is simply changing or substituting a part of your molecule to produce similar physical or biological properties. Bio meaning biology, isostere meaning same space. It's used in drug design to help medicinal chemists optimize their drug candidates. It works without drastically altering the molecular structure. You can address factors like potency, selectivity, metabolism, things like that. There are some really great reviews out there, including an article by Drug Hunter I suggest you check out. I'll put the link in the description. Nicholas, meanwhile, has done a lot of great work just reviewing different bioisosteres, explaining their pharmaceutical relevance. The lock and key mechanism of binding is kind of a primitive way to look at it. We know there are much better models for protein ligand binding. We can still use it to illustrate a point here. Traditional chemistry would say that one key and exactly one key are needed for each lock, but bioisostere replacements break that mold. So we could have many keys that interact with the same lock in slightly different ways. And our bioisostere replacement, as I mentioned before, doesn't even have to be interacting with the protein. By swapping chemical groups, we can easily tune the solubility of our compounds. We can block metabolism so that drugs last longer in your body. We can use them to explore new intellectual property space and avoid patents. We can use them to remove problematic chemical groups to reduce toxicity. We can improve the selectivity and target specific proteins. Breaking out of flat chemical space provides a great opportunity for improving the solubility of your compounds and the selectivity toward its target. There's a great article in Nature Reviews Chemistry that discusses replacing benzene with cubane and other non-classical bioisosteres to increase its sp3 character. I suggest you check it out. In 2013, researchers at Amgen reported a structure-activity relationship study on a series of indole and aza-indole scaffolds as CDC7 kinase inhibitors. Shown here is a torsional energy scan around the highlighted dihedral angles. I reproduced these results using constrained minimization quantum mechanical calculations. These scans help explain the roughly 5-fold increase in potency of compound 1A compared to compound 1B. The preferred syn conformation of the binding pose is indicated by the black arrow. For compound 1A, the scan reveals two symmetrical minima corresponding to the syn and anti conformations. In contrast, for compound 1B, a local minimum around 30 degrees lies more than 6 kilocalories per mole above the global anti-minimum. This is caused by NN repulsion within the aza-indole ring system, an effect that arose during their nitrogen walk around the 6-membered portion of the bicyclic core. Although the observed potency difference between 1A and 1B is smaller than what you'd expect from a 6 kcal per mole strain energy, this example illustrates the power of bioisosteric replacement. By controlling conformational dynamics and pre-organizing the ligand, you can improve binding affinity and reduce the entropic penalty upon binding. Okay, so picture this. In the late 1970s, there was this drug called cimetidine, brand name Tagamet. It was a game changer for treating ulcers. People who had pain for years finally had relief. But the thing was, there were some pretty annoying problems with it. Take a look at this ring right here, the imidazole ring. It was causing all sorts of trouble. Patients were coming in with weird side effects, like guys were developing breast tissue, people on blood thinners were having serious interactions, and doctors had to completely recalculate doses for heart medication. This imidazole ring binds to something called cytochrome P450 enzymes in your liver. These enzymes are like your body's garbage disposal for drugs. They're responsible for metabolism. When cimetidine gunked up the disposal, other drugs couldn't get processed properly, so they'd build up to dangerous levels. Scientists at GSK had a brilliant idea. What if they could keep everything that made cimetidine work, but swap out the problematic imidazole ring? They tried dozens of different rings, and then boom, they replaced the imidazole with a furan. Seems simple, right? Just switching one 5-membered ring for another, but the results were mind-blowing. Now you might be thinking, wait, chemists have been doing these bioisostere replacements for a long time. Cimetidine to Zantac happened in 1981, way before modern computational techniques came along. So what's the big deal about computational chemistry anyway? Here's the thing about those old school chemists, and even chemists today. They're legends, absolute legends. But they're basically flying blind. They'd make a hypothesis, spend a few months to synthesize a handful of compounds, then wait on the testing and hope for the best. It's like trying to find a specific house in a city at night, without a map, without a light, walking around. Let's put this in perspective, okay? When they developed Zantac, they synthesized and tested over 200 molecules over the period of 4 years. Today, we can generate 10,000 or more molecules in about 30 seconds. But it's not just about the speed here. Computational chemistry lets us actually see why these molecules may or may not work. We can simulate how each bioisostere might bind to the protein. We can calculate the binding energy, predict how quickly and where it'll be metabolized, even estimate side effects, all before making a single molecule. With computational tools, we now have a systematic way of exploring chemical space. What used to require $1 million licenses from software companies and supercomputers can now run in minutes on your laptop. RDKit is free, it's open source, and I can share some simple examples about how to use it for what we've talked about today. Drop a comment below and reach out if you'd like me to share these scripts with you. If you found this video valuable in any way, please consider liking and subscribing. It really helps the channel a lot. Until next time, keep exploring, keep coding, keep asking questions. Thanks for watching. See ya.